Good morning or good afternoon, everybody. Thank you so much for attending Applied Stem Cell webinar today. So today I'd like to introduce to you our TARGATT technology, which is a rapid site-specific gene integration technology. And also like to discuss the technology application in three main products and services that we provide. The first application is target CHO cells for bioproduction. And the second is mammalian cell-based library for antibody screening. And lastly, I'd like to discuss our naive IgG library system. So like I said, TARGATT technology is a gene insertion technology. So the main features include that the TARGATT technology, we can control copy numbers. So it's a single copy insertion. And it also has very high integration efficiency, up to 40%. And also it provides consistent expression across constructs because the insertion happens in a defined genomic safe harbor locus. And the expression of the transgenes usually ranges from medium to high protein expression. And the cell lines are stable because the integration is permanent. It's in the chromosome in a site-specific manner. So the application of TARGATT technology in CHO production is the following. So we have generated TARGATT CHO Master Cell Lines. And these cell lines contain the target landing pad in a safe genomic harbor locus. And then the cell line can be used to insert any genes of interest, to express any genes of interest. So the main features of our TARGATT CHO Master Cell Line is that it provides very high efficiency for gene insertion, and it's stable, and a single copy insertion, and the insertion happens in a safe harbor genomic locus. So the figures provides some data that we have done using GFP as a reporter to measure the integration efficiency. So the top panels are control. So that is from random integration. So the left one is bright field image. The right one is GFP expression. And the bottom two figures are using the integrase, the TARGATT technology. And as you can see that after enrichment, we see over 90% of the cells contain the GFP gene integrated in the desired locus. So this is just a FACS sorting to show again, before any drug selection, we were able to achieve about 13% of gene insertion. And post-selection, we were able to achieve over 97% gene insertion efficiency. And this experiment shows that the insertion using TARGATT technology is single copy insertion. So we have two different donor plasmid. One contains the green fluorescence protein, and the other contains M-cherry that gives red fluorescence. And we mix those 1-to-1, then transfected into a TARGATT Master CHO cell line. And so we were able to see either green cells or red cells. There's no overlap, indicating that the integration is in single copy. And one thing that I have mentioned a couple of times is safe harbor locus. So the insertion site is very important. And this particular benchmark insertion site is H11. And this site was originally isolated or screened in a mouse genome. And so this particular site H11 is identified in an intergenic region between two highly expressed genes. And the insertion at H11 sites results little or no phenotypic change, and it does not affect any endogenous gene expression or function. And the expression of the transgene that's inserted at this locus is usually ranges from medium to high protein expression, largely dependent on the promoter that is put in to drive the transgene expression. And we have identified orthologous sequences of the H11 locus in many different cell lines or organisms. So we have identified in human cells, HEC293 cells, iPS cells, and then CHO cells. And we also done in animals such as mice, pigs, rats. And so this site is very well validated across the species in different cell lines. So this is a testing result using our CHO master cell line for production of antibodies. So this is a collaboration with a partner, and we have two antibodies, antibody 1 and 2, and they were inserted, the transgene expressing these antibodies were inserted into the safe harbor locus H11 in the master CHO cell line. And the copy inserted is a single copy of the transgene, and the cells were grown in a 17-day fed batch shake flask, and then the bulk was enriched. So the measurement is done on a pooled cells. So we were able to see a production of over 0.5 gram per liter in a bulk enriched pools without any optimization done to the process or any culture conditions. And so this just shows the data that were measured at day 11, 14, and 17 using HPLC titering. And we see a nice increase of expression at the end of day 17, and was able to reach, some clones were, some pools were able to reach to 1 gram per liter production. And like I said, the locus is very important. So we made some effort to search for other loci in the genome to see if we can find even better site than the H11 locus. And so these shows, graph shows that we were able to find another site, ASC2, that outperforms other loci and H11 loci, a locus in expression levels and expression stability. So the left graph is an experiment using GFP marker as a readout, and there we looked at 5 different docking site location, so ASC2 and various other ones. And then the right graph, we used luciferase as a reporter to measure expression quantitatively. And so the GFP experiment we did over 82 days to look at how stable the expression lasts. And the luciferase we looked at 56 days. So from these experiments, we're able to see that ASC2 allows the highest expression level, both in GFP and luciferase expression. And also the expression is stable. So from 21 days, 42 days through 82 days. So the cell lines are stable. And then we also looked at some more relevant proteins. So this is an example of expression of an Fc fusion protein. So this is the donor plasmid that contains the Fc fusion, and that's driven by CMV promoter. And we started cell expansion at day 0 and did transfection at day 7. And then seed flask at day 27, then measured protein yield at day 33. So which is 6 days in the flask, shake flask. And so here we compare ASC2 with H11 locus. And so this is the expression level, so 142 milligram per liter after 6 days. So ASC2 has a much higher expression level compared to H11. And we also see that there's not much variability from clone to clone. So these are 20 different clones, and they all have very similar protein expression level. So now I'd like to move to using the target technology for mammalian cell-based library screening. So the traditional way of doing library screening is using either bacteria or yeast phage display. And these systems are more cost-effective and fast. And however, bacteria lacks post-translational modification. And the yeast, even though it does have some post-translational modification, but it does not contain all translational modification that mammalian proteins need to have. And then, of course, mammalian system is really the closest to full human environment, and it has all the post-translational modifications. However, the current available system is slow and laborious and costly. So we have developed the target system to really to do mammalian screening libraries. So these are some current available methods in comparison with our target technology. So basically, the black bars are the current available mammalian library system for screening. And like I said, it does reflect the mammalian cell does provide more closely to what happens in human system. However, the drawback is library sizes. So in most cases, the library coverage is very low. The highest that we can see here is about 1 million. And compared to bacteria or yeast, so this is more in the 10 million to 1/10 to the 9th size range. So our goal is to use target technology to be able to hit within an order of magnitude similar to what bacteria or yeast library can achieve, and also not growing insanely large number of plates, right? So so that's our goal, and we have achieved basically reached to a efficiency that will allow us to do that. So I kind of mentioned that already. So this is just a summary of the advantage of using target for mammalian display system. And so the main thing I want to really emphasize again is that it gives site-specific gene insertion, so that really provides a consistency in terms of expressing your target protein. So it's once you have a cell line established, you don't have to worry about if the next gene of interest is going to express differently, significantly differently. You don't have to start from your seed bank again, right? So and then it gives very like I said, it gives consistent expression. And another advantage of a target is really the size of insertion. So the phage display or mammalian display system usually has some kind of limit on the size. And a target is really relatively less limited. We've been successfully insert over 20 kb fragment in a very reasonable efficiency using target system. And then also another thing is the copy number. So we can control the copy number, which is single copy. And so this gives more control on the ratio, right? So basically each cell will express a single copy of the transgene. So so this is the system that we have for target library screening. And so basically we use a split cassette selection screening system, so that this really gives very clean result, almost without background. Right? So basically the idea is to separate the promoter and the transgene. Okay, so the promoter here is on the chromosome. Okay, so this is pre-inserted on the chromosome at a safe harbor locus. Okay, and so here is EF1 alpha promoter. And then on the donor plasmid, the reporter gene here, we use mCherry as an example, has no promoter, so it will not express by itself on a donor plasmid. The only time mCherry will be expressed is if there's a site-specific gene insertion at the safe harbor locus, right? So that will bring the promoter next to the transgene. So this will drive expression of, in this case, a red fluorescence protein. Right? If any other events that happens, we will not see expression, either random integration, there will not, basically this mCherry will still not have a promoter. So there will be no fluorescence expression. So that's really provides a very clean method to screen for library. So the efficiency of insertion is very high compared to any other mammalian system that is available on the market. So here we show that in HEC293 cells, and we were able to achieve about 44% with our target integrase system. And after enrichment, we were able to reach above 90% of the cells that contain the insertion. Okay, so this is really basic. That's the bottom line why our system is so much more efficient compared to other available method. And we also have the library screening system in CHO cell background. So these CHO cell lines also contains the same mechanisms, the same cassettes. And the efficiency, although in CHO cells, is slightly lower than HEC293 cells, but we are able to achieve 18% before selection. And post selection, we're also able to achieve over 90% insertion. So this is just a comparison of using the target screening system and the projected library size that we can cover, right? So these are molecular. So basically, this is the bacteria. I'm sorry, this is a bacteria and a phage system, and what library size they can cover, right? So I mentioned this earlier. And then here we're talking about flip-in. They compare to target system and some other system here. So the blue ones are a current existing available mammalian system. So apparently our system is way high efficiency compared to the current available system. So this is because the integration efficiency I already mentioned. So the efficiency is 1,600 to 4,000 fold improvement over flip-in, for example. So basically we're talking about 40% integration efficiency versus this 0.01% efficiency using flip-in. And if we match the cell counts that are used in these other systems, the coverage of library using target system in HEC293 cells will reach the library size that used in bacteria. Okay, so which is 1 × 10⁹ size. So now this is the last part that I will discuss today, is that we're using target system technology to build a naive IgG library for antibody discovery and screening. So first, we built the donor DNA library. So this is a naive library. So the source of the DNA is from non-immunized human donor. So we got a mix of spleen, lymph nodes, and bone marrow. And then we extracted RNA and made cDNA. And then both the heavy and light chain were amplified by PCR. And then the library DNA was assembled to donor vectors. And then we did QC, the library QC by sequencing, so both Sanger and NGS. And part 2 is to make the DNA library into the HEC293 cell library. And so in this part, we transfected the library donor DNA plus our target integrase vector and transfected HEC293 cell, the master cell line, the one that has H11 locus, and with the EF1 alpha promoter on the chromosome, the split cassette system. And after 40 hours, we did drug selection. And then we look at the QC, basically we QC the library quantity and quality analysis by Sanger sequencing and next-gen sequencing. So this is just a representative data to share with you. So our data shows that we have very high library diversity coverage and no repeat sequence was detected. The top panel here, top row is for light chains and the bottom row is for heavy chains. So we look at all these sequences, basically seeing no repeats. So it shows very good coverage of the library. So again, so this is just a summary of using our target system for library screening. And so compared to other available systems, so target, we have both HEC293 and CHO cells for library screening. And so we have a specific locus, which is H11, our benchmark locus. And the recombinase that we use is serine integrase. One of the main reasons for target to be so efficient is irreversible. The reaction is irreversible. And efficiency of gene integration is 44% in HEC293 cells and about 18% in CHO cells. And then we use a split cassette system, so which is very different from those other systems. I emphasize that this is important because it gives very clean result with almost no background. And then another advantage of using target system for library screening is its coverage. And we will be able to reach an estimated 10 to the 7th, 10 to the 9th coverage, similar to what bacteria and yeast can offer. So just to summarize, so today I presented mainly using target technology in bioproduction and library screening. So we have these products available. So we have target CHO master cell lines available either for making stable cell line or library screening. So the same thing for HEC293 cell master cell line. And in addition, we also have other master cell line, such as target iPSC master cell line. So iPSC, the advantage of iPSC is that it can be differentiated into virtually all cell types, such as neurons, NK, T, RPE, or cardiomyocytes, etc. The list keeps going, right? And then we also provide engineering custom master cell line. So basically, if you have your favorite cell line that you wanted to use our target technology, we can actually engineer our target system into your favorite cell line so that you can take advantage of using our target technology for your purpose. With that, I'd like to thank you very much for attending this webinar. And now I think we have some time for questions.
Thank you, Dr. Tensai. Now we will be taking some questions. The first question is a library constructing plasmid available for us to make our own library for ourselves?
Yeah, so that is available. So basically, so what we could provide is the master cell line and also the vector in which you can build your library in. And so yes, absolutely. What is the question?
The next question is, can you tell us a bit more about this screening process? Does it require clonal selection?
So the screening process, so okay, let's talk about, so if we use it for CHO, stable cell line, you have a choice to either make clones so that you have a stable cell line and basically a seed bank that you can use, go back to. So yes, so that you would be doing cloning, and which is very straightforward because the efficiency is so high. And sometimes people just wanted to do a very quick experiment, research grade, just look at expression, then you would not need to do a single clone. So you can just look at a pool. So because our system allows you to enrich the gene insertion up to 98% in CHO cells. So the pool will pretty much do the work. So in that case, you will not need to do cloning. So I can see this. Should I just, you want to read that question?
The next question we have here is, could you describe the business model to access your technology?
Yeah, or thanks for that question. So basically we have several ways of doing it. So one is we can provide service. So basically you tell us what your purpose or your goal is, then we will provide as fee for service, okay? And so that's one way to do it. And another way of doing it is we can provide a non-exclusive license to you, and so that you can do either make it yourself, or you can still use our service to do it for you. And in terms of the license, there are two types of license. One is for research use only. So that's a very reasonable license fee, so you can do it for internal research. And the other is commercial license, so that we can discuss on a case-by-case basis.
The next question we have here is, how long does it take to obtain a pool? Could this be used for?
I think it's a high throughput.
High throughput antibody production.
Okay. Yeah. So we're just reading the question while it's coming in. So sorry, we may actually have a pause here. So yeah. So basically, to get a pool, it only takes about a couple weeks. So that's very efficient. Okay. And then the other question was high throughput. Could this be used high throughput? Yeah, so we have developed a high throughput format in the plates. So we do have capability of high throughput production. Yeah, so please do contact us. So if you don't, if you wanted to pursue further, so contact us at info@appliedstemcell.com so we can discuss in more details, particularly to meet your purpose.
Okay, thank you all. Those are all the questions we have time for, but we will definitely answer all your questions individually by email. Please send them your additional questions to info@appliedstemcell.com, as you can see on the screen. We will also be sending you a copy of today's presentation along with a recorded webinar link. Thank you all so much for attending, and have a good rest of your day.